Hello and welcome. I'm Dr. Mark Brandon, an associate professor at the School of Medicine and the Department of Psychiatry at the Douglas Hospital Research Center at McGill University. As a neuroscientist, my focus is on unraveling the complex mechanisms by which our brains encode and process spatial memories. Today, I'm excited to present another installment of our publication series. This series is designed to demystify the intricate details of neuroscience research and make it accessible and engaging to all. In this episode, I will share an exciting study conducted by our lab, which was recently published in Frontiers in Neural Circuits. The paper was led by Dr. Saishree Badrinarayanan during her PhD work in my lab, and is titled A Characterization of the Electrophysiological and Morphological Properties of Vasoactive Intestinal Peptide, or VIP, interneurons in the medial entorhinal cortex. This research focuses on the role of VIP-expressing interneurons within the medial entorhinal cortex, a critical area for memory and spatial navigation. Our study utilized in vitro whole cell patch clamp recordings and immunohistochemistry to investigate the unique properties of these cells. The role of the medial entorhinal cortex is to translate one's movement into a coherent neural code for spatial location, primarily through the action of grid cells. However, these cells' functioning relies heavily on a delicate balance of excitation and inhibition within the medial entorhinal cortex. Our findings indicate that VIP interneurons play a crucial role in maintaining this balance by providing targeted disinhibition to principal cells. Our experiments revealed that VIP interneurons exhibit distinct electrophysiological properties across different layers of the medial entorhinal cortex. Interestingly, these properties vary along the dorsal-ventral axis of the medial entorhinal cortex, suggesting a finely tuned mechanisms which adjust inhibitory and excitatory influences based on spatial processing demands. Morphologically, we observed that VIP interneurons displayed diverse structural characteristics that differ significantly between the superficial and deep layers of the medial entorhinal cortex. These structural differences likely influence how these interneurons integrate into the overall circuitry of the medial entorhinal cortex, affecting how spatial information is processed and integrated. These insights are not just critical for understanding the basic neurobiology of memory, but also have implications for disorders involving memory impairments, such as Alzheimer's disease. By mapping these details of interneuron function, we can better understand the neural substrates of memory and develop targeted interventions to mitigate the effects of neurodegenerative diseases. Thank you for joining us today as we explored the fascinating role of VIP interneurons in the brain's spatial and episodic memory circuits. Stay tuned for more episodes where we'll continue to uncover the brain's complex circuitry and its implications for understanding brain function.